Right, special senses. Let's see. I would like to have you guys take out a sheet of paper and take your term list. And, ooh, look at that. Hold on. I got to figure out how to learn this again. And take out your term list. And can you guys see the heart?
Yeah.
Good. All right, quiz time. No cheating. Everybody on their own. But make sure you go with your term list because on the test you can use your term list. All right, question number 1. What's this pipe here called that's coming out of the aortic arch? The first one that comes out of the aortic arch. Number 1.
Wait, I have a question.
Yes, sir.
Didn't we do this last week?
Yes, we did. We're going to do it all the time until the final.
Oh, okay.
That way, I train you for the test. I'm just going to keep that with the way I had it in the class because I want to make sure everybody's a little bit studying this stuff all the time. So
okay.
If you have your term list and then you go through, by the time you get to the final, if you understand these things, you're probably going to be fine. All right?
All right.
All right. Number 1, give me that first thing that comes out of the aortic arch. Number 2, give me the name of this flap that I'm pointing to. All right. Number 3. Give me this chamber that I'm pointing to. Ready for number 4? Number 4, you see the blood vessels, the blood things?
Yeah.
Good. Number 4, what is it pointing to? Good. Nobody's speaking out. That's good. Number 5. Anybody tell me if I'm too fast. Just holler out, I'm too fast, okay? Number 5, give me the name of this white blood cell type. Number 6. Give me the name of this gyrus. Number 7. Give me the name of this lobe. Number 8. Where about I drag that with me here? Hold on. Number 8. Give me the name of this structure. I gotta get better pictures of this stuff. What am I at? Number 9. Let me see, I gotta drag that down. Sorry about that. Give me the name of this structure there. I know I'm mean today. Sorry about that. And then number 10. Give me the name of, hold on, I go to the next one here. Spinal cord. You see the spinal cord, right?
Yeah.
Good. I'd like you to tell me the name. Oh, why is there the names on it? That's dumb. That was me bad. All right. I'd like you to tell me. Oops. The nerves. Oops. Sorry about that. Yeah. The nerve things that it points to here. That's number 10. Number 11, I want you to tell me that yellow swelling that it is pointing at. That's enough. All right, good. Let's solve that. Let's go to the heart. There it is. Everybody see a heart?
Yeah.
Number 1 was, what was that? Anybody?
That's a brachiocephalic artery.
Good. Brachiocephalic trunk. But
Trunk,
good.
sorry.
That's okay. A trunk is often used as a name as a vessel that is an artery that splits really fast after it comes out of it. So the brachiocephalic splits about up here probably, and then a branch comes out that goes down and one goes up. That's why they use that term. So just to clarify that.
Oh, cool. Okay.
Yeah, hey, wherever you find patterns in anatomy, you're going to go for the patterns. And then you use them until you think they're wrong. That's how I do it. All right, number 2 was, somebody tell me number 2. Yes, Dennis. Anybody?
Atrioventricular valve.
Atrioventricular valve. Perfect. Good. There are one here and one here. And then number 3 is this chamber here. What was that called? Anybody? Nobody?
Is it the right atrium?
Yes, thank you, Carlos. It's the atria, the right atrium. On the list, it says atria. That's good enough, but more specific, it's the right atrium. Atrium is a singular, the word we use for singular, and atria is for plural. That's why it's 2 different words. All right, from there, we moved to the next one. What was that? Anybody?
I think it was a red blood cell.
Yes, it was a red blood cell. And then what was number 5? Anybody?
Eosinophil.
Good. Eosinophil. And then from there, we went right to the brain. Le brain. Let me just do this real quick. Sorry about that. Come on, baby. Let me copy this for it easier. All right, the first thing in the brain I was asking was this blue thing here, this gyrus. Anybody? What's that?
The precentral gyrus?
Not quite. It's the other one. Say the other one, Chris.
Primary. No.
No, stay Chris. Stay with Chris. So
Yeah.
post-central gyrus. So the one in front, if you're not on mute, go on. Anybody who has background noise on mute, please. If it's the one in the front, it's the pre-central gyrus, and there's one in the back of this line here, this dividing line, is the central sulcus, which is not a term on the list, I don't think, but it's in the booklet. And then behind it is the post-central gyrus.
Gotcha.
That makes it. So pre means before and post means behind. All right, good one. Let's go to the next one. What? Hey, background noise, people, turn off. Wait, I can do it. Hold on. Who is not backgrounding? Oh, they're all backgrounding. Okay, maybe it's mine. Okay, back to the screen here. Brain. All right, so then we ask for what's this lobe here called? Anybody?
Insula?
Insula, yes. Good. And then we went down to the front in the brain stem area. The front here, what is that area called?
Pons?
Yes, the pons. Super. And then we went all the way down to here. And what is it pointing to here? Anybody?
Thalamus? No.
Thalamus, perfect. Ooh, you got on the roll there. Perfect, good job. And then from there, we went to the spinal cord, which I made not sure. Oops, hold on. There we go. Not sure why it's showing this conomedullaris, but anyway, it's for some reason I made a mistake there. Oops. Anyway, so then I asked for number 10. What was number 10? Right here. Anybody?
Do you mean the green thing?
What? Say it again, Alice.
Do you mean the green thing?
No. Well, we could do the green thing too. Actually, that's a good point. But that's not that's not on the list. The nerves that come out of the spinal cord as the spinal cord terminates. What are those called?
Cauda equina? No.
Cauda equina. Perfect. Dude, you're really on a roll. That's super. You studied, huh? Good. And then number 11. What's this yellow bulge called? Anybody?
Sensory neuroma?
Nope.
Or is it the dorsal root?
Yes. Yeah. Say Dennis. Give it to Dennis. Say
okay.
Dennis.
The
That's all right.
dorsal
That's all right. Check it.
Yeah.
What do you say, Dennis?
The dorsal root ganglion.
Dorsal root ganglion. Perfect. You guys are great. All right. So these are getting a little bit more complex. In this chapter, we discussed the nervous system in more detail. We will spend most of our time covering the special senses, but start with some general concepts. One of them is the fact that we differentiate between awareness and sense delivered to the brain. Somebody's not on mute. Get on mute, please, everybody. Thank you. All right, where was that? One of them, in fact, that we differentiate between awareness as a sense delivered to the brain and having it be interpreted for meaning. This is actually often discussed in modern psychology when we try to understand suffering. That is a significant portion of suffering that comes from interpreting a stimulus such as pain and not simply having the awareness of pain. What terminology is used to describe the two? Choose both.
Clearly, sensation and perception.
Good. Sensation and perception. That's pretty clear, right? As an information piece?
Yes,
Yeah.
it is.
Good. Let's go to the next one. If questions arise, just raise your voice and we'll go to the topic. But otherwise we'll move forward unless I think it has some importance to it. Sensory receptors are found throughout the body. They inform the brain of different types of stimuli. Choose all receptor types that apply.
Nate.
Jessica, all
Nate?
of them?
All right, I heard Nate first. Nate?
1, 3, and 4.
Thermonocy, mechano. What about photo?
All of them.
4 is also.
Well,
Oh yeah, I said 1. I said 1, 3, and 4.
Oh, 1, 3, 4. Nosy is also, nosy means pain. So actually they're all correct. Or they should be all correct. Nociception, I put that in there because nociception actually, when you interpret that as a word, it means danger receptors. But for us humans, the way we interpret that is the pain. That's when you twist your ankle, your nociceptor fire, off that you twisted your ankle and you better don't keep walking like that.
Yeah.
So that's one also. All right, so they're actually all correct. So that's perfect. And when you go to the slidey, now where's my slidey stuff? Here's my slidey. There we go. We have different stimulus types right here, all of them. Good. I know the nosy is kind of a trippy one because it's not that easy to understand that. All right, good one. So we classify receptors by what they code for. Some nerve endings are simple, such as free nerve endings, or onion layer looking like nerve endings. Which are encapsulated ones, but while other ones are even more complicated. What senses do the complex receptors transduce?
Cesar.
Cesar.
Vision, hearing, smell, taste, and touch.
Good. So the complex ones aren't the touch. No, not touch. Which one you say?
Touch.
No, touch is not. It's taste, vision. Say it again. What do you have?
And hearing.
Cesar.
The first.
No, Cesar.
Oh.
I know. Hello.
Uh-huh.
Say it again. Which ones do you have? So I didn't listen right.
I put vision, hearing, smell, taste, and touch, but I didn't get all the points.
Yeah, so you did vision, hearing, smell, taste. Touch is not one of them. Touch and pain are not. So these first four. So that's Dennis. Yeah, you were correct with that. Just want to make sure we discuss it too. Yeah, that's probably one you got wrong. You can go back and get all the points if you want. Well, you should. All right, so that's good. So number 4, many nerve stimuli don't need to excite our brain all the time at all times. It often is more important to register change and not continuously be made aware of a specific temperature, for example. Often the water temperature one likes at the beginning of a shower is cooler than at the end of the shower because one gets used to the ongoing stimuli. Other senses such as touch or pressure or the brightness of light also adapt. Can you name 2 receptor types that do not adapt?
Jessica, the last
Jessica.
Two.
The last
Jessica.
2.
Say it, Jessica.
Last 2. Proprioceptors.
Well, you gotta speak them out. Speak them out. We say.
Where is it? Proprioceptors.
Proprioceptors. And proprioceptors. And what's the other one?
Nociceptors.
Nociceptors. Good. See, that's the cool thing about these complicated words is you can just say them the way you read them. You spell them out, and you're gonna be fine. It's different than English. English is complicated about that.
Yes.
Like I have a hard time reading English stuff sometimes, how it sounds. But this stuff is Latin, so you just say how it reads, and then you're good. All right, super. No questions to that? I move forward. Otherwise, speak up. When we discussed the primary sensory cortex in the brain chapter, we mentioned that the distribution of nerve receptors is not equal for all body areas. For example, the lips or fingertips are much more densely innervated with sensory nerve receptors than the back. We can feel a coin in our pocket and know what size it is. Try doing that with your back. How big an area that one receptor innervates is called a receptive field. What test can we use to determine the size of a receptive field?
Dennis, the 2-point discrimination test.
Super.
Mr.
Yes.
I got the quiz.
Wait, Dennis, go on mute. And who talking? Shirley?
Shirley.
Okay, you were talking. Well, just what were you saying?
That I got the answer wrong on the quiz. The 2 determination test.
Yeah, and you thought you got it right?
Yeah.
Okay, let me write it down, then I can look at it. And spell it. If you do it again, spell it exactly like it's in the book. But I'll look it again at all this. Do you remember how you spelled it?
I think it's the same way that it's on
Okay.
the.
I'll look it up after class. If you want to stick around, you can. Otherwise, I'll just look it up in the background. I took some notes here.
Okay.
So the 2-point discrimination test is basically taking, you know, you can even just take a wire basically and well, not too pokey because you're gonna hurt them, but see how far apart can you stick 2 wire pieces, and the person you do it to thinks it's 1 point that you're touching, or if it's 2 points that you're touching. And in the back, you can go fairly far in the fingers, never mind. And so that's what that code's for. All right, moving right along. In trying to understand vision, we have to mostly study the eyeball. The eyeball is a sphere that transforms the visual world we see into nerve impulses, which is the electricity, which that is the language of the brain can understand. It has light coming in on one end and a nerve or cable leaving on the other. Inside, a clear picture of what we see has to project into a tiny screen at the back of the eyeball. There, a gazillion receptors get activated when projected on. They then send a signal to the brain when it appears as pixel projected onto a primary visual cortex of the occipital lobe, sort of recreating the visual image. Let's now consider the parts of the incredible organ. First, can you match the function of the different layers or tunics? No, anybody?
Me.
Chris?
Alice.
I think it's the middle one.
Sorry, Chris. No. What did you say, Fibers?
The middle one.
Speak it out.
I can't pronounce the first
Sclera.
one.
Sclera.
Sclera and cornea.
Yes, sclera cornea, vascular tunic.
Iris. I can't.
Ciliary. You
body.
just have to slow it down. It'll work out. Ciliary body and choroid.
Choroid.
Choroid. There you go. And third.
Pigmented layer and neural layer.
There you go. Super. All right. Those are just anatomy terms. If we then look at a cross-section of the eyeball, we can separate the area behind and in front of the iris or the lens. Hold on. Oopsies. Iris or the lens. The larger space behind the lens, known as the posterior chamber, consists of a jello-like, somewhat firm but wobbly substance called the vitreous humor. It helps the eyeball structure's integrity by pressing outward against its wall. In the front chamber, a more liquid aqueous humor provides the internal pressure and also brings nutrients to the eyeball. Nutrients get used up, so this fluid has to be secreted from the bloodstream and drained in order to not accumulate and exert excess pressure onto the eyeball. Can you name the place where the aqueous humor drains from?
Chris?
Chris?
The canal of Schlem?
The canal of Schlem or Schlem, yes. This must be a German word. The German language, the SCH, they do zh, but Schlem is perfect. All right, that's just an anatomical term. Let's keep going here. The eye sees a picture best when projected to a specific area on the retina that is most densely populated with receptor. In order to do that, the eyeball has to be able to move freely around its own axis. It does that by having 6 muscles attached onto it and pull in all possible directions. The 4 recti muscles enable us to look up, down, and to the sides. What muscle pulls the eyeball up and out?
Carlos.
Carlos?
Yeah, and inferior oblique.
Super. Inferior oblique. That is correct. I need to go to that picture because I love that picture. Okay, so we have a vision. We have a few things here. We have a few layers described. And then, here are the layers, the tunics. And then at some point here, we get to the eyeball. This is the canal of Schlem here as it drains out. So that's the front part, the aqueous humor, the vitreous humor in the back. And then here are the eye muscles. And so, well, I guess that's, in the lab, that's always a big piece, but basically they're all attached in the back of the orbit in the bony ring or in a ring, and then they come and attach to the eyeball from there in different directions, and 4 are just going right straight up, and they're called rectus. Rectus means straight. So you look it up and the superior contracts, you're looking down when the inferior contracts, and so forth. And then 2 of them one comes above and has a little hook that it moves through it and then attaches to the eyeball. So when that one contracts, the eyeball sort of gets pulled this way, so the eye actually looks down and out. And the inferior one is attached like here and comes into the eyeball from this angle. When that comes down here, it somehow, the way it moves the eyeball up and out. So that gives us pretty much all the movement we need. So that's pretty nifty. Let's now understand the transformation from light stimulus into a nerve impulse. The whole process boils down to molecules stacked up in round discs changing shape when they are activated by light rays. There's 2 types of photoreceptors. One of them is so sensitive that it picks up dim light. Those get overwhelmed when light is too bright and they stop firing. We feel the process of them bleaching out, that's the term they use, when we go from darkness to bright light quickly. It kind of hurts. The other receptor types activated when it is light. They come in 3 different subtypes that help us see color. What are those one called? What's that one called?
Jennifer Cones.
Who's that? Jennifer?
Jennifer.
Coins. Yes, super. So what do we need to know about? The cool thing about this is it stacks these discs up, and this is the chemical transformation between light hitting it and light not hitting it. So that's a simple what it does molecularly, and then that changes the molecule enough that we fire a nerve impulse. So that's kind of crazy, but that's how it works. And then we have the rods and cones, and you feel that every time you go from dark to light, you get bleached out and it hurts, and from light to dark, and you can't see nothing for a little bit, and then you start seeing better and better. It takes about 20 minutes to change from light to dark, so all the rods are activated to be helping you see, because they're still bleached out, so it takes a while to accumulate the molecules that way. So that's what that is. There's a part in our peripheral vision that projects onto the retina where the optic nerve leaves the eyeball. That area, called the optic disc, has no photoreceptors and cannot transduce the picture. The brain actually just imagines that part of the visual field. On the other hand, the area of the retina coding for the center of our visual field is most densely filled with receptors, giving us the best, most acute vision. What is that area called? Anybody?
Dennis, fovea centralis.
Good. Fovea centralis. Good, super. And again, you stop me if you have a question, and do not hesitate to ask, please. I welcome questions. Light rays entering the eye have to bend, we call that refract, in a perfect way that they reunite on the retina sharply. If not, we see blurry. The lens can change shape, which changes the angle of the light rays that the light rays bend. It can accommodate for images at different distances. The rounder it is, the steeper the angle it bends. The light rays does a round lens best focus images that are close or far away. Anybody who hasn't spoken yet?
Kirsty, close?
Yep. Close. Yes. I always have to look that one up. But it's close. The more curved, the more close the object. We need reading glasses. That was Kirsty. We need reading glasses as we get older because the lens becomes less flexible, which makes it bend less. So the closer objects are less accommodatable. Some of us also need glasses because it is very delicate to project the perfect image onto the retina. In some of us, the light rays reunite not quite on it, but either a little or in front, or behind it. In farsightedness, the eyeball is short and the image gets projected behind it, making us see close objects blurry. Is that true or false? Somebody who has not spoken yet.
True. Melissa?
Good. There we go. Farsighted. That's true. All right, next. Good, good, good. Thank you, Melissa. We can understand the sharpness, the acuity of one's vision by measuring how well they can differentiate 2 points at a distance. What tool do we commonly use to test the visual acuity? Anybody who has not spoken? Come on, I got a couple left, I believe. No? Catherine or Ayana there? Or did I not take notes? All right. If not, open for everybody. Round 2.
Snellen eye chart.
Snellen eye chart. Dennis, you are on it. That's super. Super, super, super. Let me take that down. Good. Okay, so that's just a tool. That you do that in the doctor's office. You just look at these letters and they get smaller and smaller. That's basically what that is. Interestingly, we can see 3D. This is made possible because we have 2 eyeballs that mostly pick up the same picture, just from a slightly different angle or perspective. If you overlay those pictures, voila, we can see three-dimensional. It's also just cool to have 2 eyes. In case one don't work, we can still see. In order to be able to overlay the information from the 2 different eyes, both stimuli have to be processed in the same area of the primary visual cortex. And that's back in the occipital lobe, in the back of the head. This is accomplished by having half of the nerves cross over to the other side and join that side's optic nerve. Now, both impressions of a visual field can be brought to the occipital lobe for processing, which I think is very smart. What place do we call that where half of the nerves cross over?
Jessica, optic chimes. Chimes. I
Optic
don't know.
chiasm. Good.
Yeah, that
Optic chiasm. Yeah, that's a very weird word. And that's like underneath, right here, underneath it, further down is where the sella turcica is. So if you remember the skull, it's right above that place because it's right above the pituitary gland. Tears are great. They clean and nourish the eyes, nourish our eyes' cornea. They also help us process emotionally. Crying stimulates the release of endorphins. For example, what do we call the tear glands?
Chris?
Clearly, lacrimal gland.
Clearly, lacrimal gland. Perfect. So that's the tear gland. When we see an ear, we likely associate it with the sense of hearing. We also have the sense of balance and equilibrium in that area. In which part of the skull do we find the inner ear? Round 2.
Petrus portion.
Petrus portion. That's Alice, right?
Yeah.
Good. And again, if anybody has a question, you speak up. Our ears pick up sound waves that travel through the funnel-shaped external visible ear. They then vibrate the eardrum or the tympanic membrane and pass the vibration on to 3 small bones, which conduct the waves to the organ of corti, or hearing. Organ of hearing, the cochlea. Can you choose the 3 little bones or ossicles in the middle ear?
Chris, Malleus, Stapes, and Incus?
Yes, Malleus, Stapes, and Incus. Super. And again, those are the smallest bone in the body. And you don't have to study them for me. It's just what's interesting is that fact that we have the sound wave come in. You can see this, yeah, right? Because sound wave come in, the vibration of the eardrum, and then these ossicles vibrate, and then we vibrate the cochlea, which is the snail-looking thing that then makes electricity out of the sound wave. And sometimes when we get loss of hearing, that's because those little ossicles get sclerotic, so they get they get a little arthritis basically in them. All right, number 18. The inner ear is basically a fluid-filled channel system carved out of bone. The snail-looking cochlea makes electrical impulses out of sound waves. the three half circles or semicircular canals and their base, called vestibule, encode for starting and stopping motions in the different directions, which helps us with balance. Within the bony channels, we find membranous ones filled with a different fluid. In the cochlea, the bony labyrinth, together with the membranous one, creates three such channels. The top and bottom one connect to the snail's apex as it narrows. What are the three channels within the cochlea called?
Kirsty, scala vestibuli,
Yep.
scala tympani, and
Yep.
scala media.
Super. All right. Scala must mean chamber or something. I did not look that up, actually. But what I always find fascinating is that, where are we here? That's probably a fairly decent look. So the outside here is the one fluid, and then this membrane inside of that bone. The bone is the gray, the brownish stuff. And then this membrane inside of here has a different fluid. And depending on how big the sound wave is that comes down, because the pitch is the sound wave size, right? Not the loudness, the pitch. And depending on how big the sound waves are, at some point it hits the bone. And when it hits the bone, because it's too big, it's going to cross over, go down and cross through that middle layer and basically go out the bottom layer and vibrate into space. But as it does that, it vibrates these hairs here, this gel thing inside. And when these hairs vibrate, they fire a signal off. So that's basically, hearing is hairs bending. And when the hairs bend at the right place, they fire the signal. And then this nerve part specifically goes right to that part in the brain that hears whatever that sound level is, whatever that pitch is, if it's a deep voice or a high-pitched voice. That's basically what that is. That was pretty clear, right?
Yeah.
Yeah.
Good. Good. I need something. All right. The middle, almost done. Look at that. We're doing pretty good today. The middle of the three channels houses the organ of Corti, or the organ that makes nerve impulses out of sound waves. Its basic structure consists of a bottom membrane, which gives the rise to specialized hair cells that have cilia sticking up, touching another membrane. When the sounds waves, which are the pressure waves, travel through the blah, 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 I just talked about that. When they vibrated, thousands of nerve cells are spread along the entire cochlea. The ducts of cochlea narrow at the circle to the apex. What is the place at the apex called that they connect?
Helocotrema, Melissa.
Helocotrema, Melissa. Super. And look at that. A couple more. The hair cells place along the cochlea determine the pitch we hear. The fact that the duct narrows as it reaches the apex makes sound waves of different pitches or different heights touch the bony wall, different locations along the snail. Wherever a wave touches the bony wall is where it crosses over to the organ of Corti. Then it vibrates the hair cells in that location. Do high pitches cross over at the base or at the apex?
Jennifer, base.
Jennifer, base, yes. Hires are up and down higher, so they go over fast. The part of the inner ear that helps us with balance essentially picks up any motion our head does and translates it into nerve impulse. Any motion starts and stops, even if very tiny. The head accelerates and then decelerates. In a total, we have 5 areas of the inner ear picking up that kind of motion. 2 deal with an up and down, as well as sideways movements. You feel those activate when you're in an elevator, especially in a long one, or in a speeding car. The other 3 encode for rotational movements through all the 3 planes. You feel those when you spin in a circle, doing a cartwheel, or doing a cartwheel. In the organ of equilibrium, hair cells don't bend due to sound waves when they displace them. They bend because they are held in place by a weighted membrane that lags behind, so to speak, when our head moves. What part of this organ registers horizontal movement for us? Oh, I got one more. Horizontal movement, what part does that? I always have to look it up. Nobody knows? There we go. Auricle. See that? Did nobody get that right or just not speaking up?
I got it wrong.
You got it wrong? Just because, or is it understandable?
It was
You can go over it.
Huh?
You can go over it.
Well, the way I look at it is basically I got the bottom. Then I look at, where is it? Come on, give me some better picture of this stuff. Still not. When I look at this here, the snail we talk, that's the hearing. Then this whole thing is equilibrium. And at the bottom, we got that vestibule, and that's the auricle and the saccule. There's two of them in there. And one goes for up and down motion, one goes for sideways motion. And then we got these semicircular canals, and those are for rotational movements like cartwheel and so forth, like that, or the airplane, or however we want to visualize that. And inside that base, we have that auricle, and that's horizontal movement. That's when you're in a car going fast or slow, or in an airplane, when you start the airplane or slow it down. And then the saccule encodes for vertical, and vertical will be the elevator motion. And so that's basically just words. What's more important for us is that you understand what it does, because it's not on the term list for the anatomy. All right. That okay?
Yeah.
So that gets us to the last topic, and that's taste and smells. And that makes us appreciate the world of odors. The mouth, especially the tongue and the upper nose, have receptors that detect chemicals. Tiny cilia or hairs in this area don't react to movement or sound waves, but to chemicals that attach to receptors in their membrane. Go on mute, everybody, please. Our sense of smell is limited in compared to other animals, but even though we pick up thousands of different fragrance combinations, our mouth, however, only responds to 5 basic tastes. Can you name them?
Carlos.
Yep.
And it's bitter, umami, sour, salty, and sweet.
Super. Not creamy and not spicy.
Yes.
That's a pain receptor, I think. All right. With that, and everybody knows umami at this point in life. Do you get the umami?
I never heard that before.
That's a good one, right? When I learned anatomy, it was not a taste. And then they added it, and so it's fairly fresh that they realized the MSG, for example, that makes food taste more flavorful. Those things, that's umami. That's that flavor. So it's the flavor taste bud, so to speak. I know, it's interesting stuff. All right, how are you doing with that? Seems everything is pretty okay, right?